Hello and welcome to this Lupus Forum podcast on the cGAS STING pathway. This should be a really interesting and informative discussion about the emerging role of cGAS inhibition in lupus. My name is Ed Vital. I'm chair of the Lupus Forum and professor of translational medicine at the University of Leeds. And this is a pathway that I'd only really, I think, started to understand relatively recently compared to other things I knew about lupus pathology. But actually, I think it has a really fundamental role in the disease. It helps us understand how this disease happens. And it's also really timely now because there's a lot of new therapeutic approaches being discussed about this. So I'm really glad to be joined today by somebody who's done some really innovative work explaining how cGAS fits into lupus pathogenesis, and that's Professor Michelle Kahlenberg. Michelle's a professor of internal medicine and dermatology and vice chair of research for internal medicine at the University of Michigan. Welcome, Michelle.
Thank you, Ed. It's my pleasure to be here.
So we've both worked on some similar ideas, haven't we, about how interferon activity in early stage of disease and in the skin works in the pathogenesis of the disease, and I think this pathway, as you've recently shown, seems to play a really central role. So first, I just thought I'd take a big step back and talk in really basic terms about where this fits into lupus pathogenesis. So I always, when I first started getting into lupus, I always thought this is basically a kind of a B cell centric disease, right? So you've got autoantibodies that we measure in the clinic, you've got low complement and immune consumption, immune complexes, and we see them in immune complexes in organs, and we target B cells, and that's how we treated it. So I was very much thinking on all of that. But that view of lupus pathogenesis leaves a lot of things unexplained. And I think I always thought this is like, well, the antibody repertoire doesn't wholly, like we've got lots of autoantibodies that target lots and lots of different molecules that are structurally completely different. The only thing they've got in common is they're all found in cell nucleus. And I was like, so how does that happen? And how does the B cell abnormality arise in the first place? And then all these different clinical presentations you see, and disease that seems to be so resistant to therapy sometimes. And I think when I started thinking a lot more about the role of innate immunity and like pattern recognition that we're going to talk about today, it helps explain that a lot better to me. Because firstly, we've got that clearance hypothesis, haven't we, which says that the reason you get lupus is because cells die and aren't cleared properly. And that leads to lots of antigens being around, nuclear antigens, and lots of that antigen around, then that's why the immune system responds to that. And I was like, yeah, that explains the antibody repertoire that you get in lupus quite well, doesn't it? And then I also started to understand a little bit about how these pattern recognition receptors might just be more sensitive in detecting that stuff. And that's why some people are more susceptible to lupus, to autoimmunity than others. And that makes a lot of sense when you know about all those monogenic kinds of lupus like children get. Essentially all the monogenic lupus is always either some kind of defect of clearance of self-nucleic acids, or it's some kind of increased sensitivity to those nucleic acids. So yeah, this sort of cartoon that's on the screen now for those who are watching on YouTube or something tries to sort of show, to put that in perspective, that the way a lot of us think lupus happens now is you start off with something like you've got increased apoptosis of cells or secondary necrosis and defective clearance, which leads to lots of nuclear antigens around. And then we've got pattern recognition receptors like toll-like receptor 7 and 9, for example, that detect that. And you've got either too much of that cell death or two increased sensitivity of detecting that, all of which then converges on type 1 interferon. And we all know type 1 interferon is important now, and type 1 interferon then goes to this JAK-STAT signaling or JAK1 TYK2 signaling, and then it gives all these immunostimulatory effects like the B cell differentiation and making plasma cells and activating myeloid cells and all these other things that are downstream. And so this innate immunity is a much higher point in the pathogenic process, I think. I think that's how I kind of conceived of it now. And to me, I think that makes a lot more sense. But as we're going to talk about today, that's still not a complete picture of the pathology, is it?
Yeah, I think there's definitely more to the story. And I think what we're learning now is that there's many more contributors to generating this type 1 interferon than just toll-like receptors, right?
Yeah,
Yeah.
and to actual death of cells isn't just the
Mhm,
only way you can get antigen as well, isn't it?
mhm. Very true.
So that's where cGAS comes in, right? Could
Yeah.
you tell us a bit about that?
Yeah, so cGAS is a sensor of double-stranded DNA, and when it's activated, it's found in the cytoplasm. So it's, a lot of these toll-like receptors, they're sort of sequestered into compartments within the cell, so they can find their triggers and the nucleic acids that they bind in cytosolic compartments, or not cytosolic, but in endosomal compartments. So that's how they, get exposed to some of their viral nucleic acids and other things. But some things also get into the cytoplasm. So we have this whole cadre of sensors in the cytoplasm of the cell as well. And this has really evolved as an antiviral sensing pathway, right? When viruses get into the cytoplasm, this is why we developed this pathway so we could sense them and turn on an antiviral program, which is really what the type 1 interferon response is. That's evolutionarily why we have it, is to sort of, stop viral replication and, try to get rid of the cells that are infected. That's sort of, you
So
know,
like so like the endosomal sensors are
mhm.
detecting things from outside the cell,
Yep,
which is
exactly.
engulfed into the cell. So they're
Hmm.
sampling the outside
Hmm.
world,
Yeah.
whereas the cytosolic ones are
Hmm.
sensing things that are already within that same cell.
Sure, sure. Yeah. So viruses get in there, and that's one of the places they can, be detected. So cGAS binds double-stranded DNA and then generates signals that turn on downstream signaling pathways, particularly a protein called STING, and
Mm.
it causes STING to then turn on the signaling molecules that result in production of interferons, which then signal exactly as you showed in your cartoon. And it turns out that it's not just viruses that can activate cGAS. There's other things that can also generate DNA. Um, there's micronuclei that can be formed out of our own nuclear DNA. But the other place that there's generation of DNA that gets into the cytoplasm is from the mitochondria. And it's been known for quite some time that when a mitochondria gets damaged, the DNA can be leaked into the cytoplasm as part of a garbage disposal mechanism.
Yeah.
And if it's not regulated properly, it's left available for cGAS to bind and get turned on. And so
So
one of the,
really, this kind of part of the immune system is it's like our own nucleic acids can be just as immunogenic as viral nucleic
Mhm.
acids. It's all about them being in the wrong place.
Yeah. Yeah. And interestingly, as you mentioned, like a lot of the genetic risks. So one of the main regulators that we have to prevent cGAS from getting turned on when our nucleic acids get leaked out is a protein called TREX1, which degrades the DNA in the cytoplasm. Turns out TREX1 mutations leads to lupus-like phenotypes. Um, and so, this whole disposal mechanism for the nucleic acids is really important because when it's not regulated properly, it
So
causes
you
disease.
want your
Yeah.
nucleic acids safely inside the nucleus. You don't want them in the cytoplasm of the cell because
Yep.
that'll trigger something like cGAS, or you don't want
Mhm.
them outside the cell because that'll
Mhm.
get into endosomes and trigger all those
All the
things.
TLRs. Yeah, exactly.
So how does this
Exactly.
fit into the initiation of lupus exactly then?
Yeah, well, if we can go to the next slide, I'll tell you a little bit about the research that we just published. So one of the very interesting questions that we have had regarding, triggers of lupus and triggers of lupus flares is how the skin responds to UV light or sunlight. Because we know in lupus patients, the sun is really good at turning on rashes and
Mhm.
sometimes can actually trigger systemic disease flares, meaning they can get like kidney inflammation or nephritis. They can feel very physically ill when they get exposed to the sun. And this is different than a lot of other autoimmune diseases where we actually use sunlight to treat the disease, like psoriasis and eczema, for example. We put people out in the sun and tell them to get sun exposure because it helps their rashes. But in lupus, if you do that, it makes people sick or makes the rashes worse. So we were very interested in this idea. We term this as photosensitivity, right? You're sensitive to the sun. Um, and how the UV response in lupus skin might be abnormal. And it turns out we've known for a long time that UV can actually trigger um, oxidation of the DNA within mitochondria. And it does this. We're not really sure exactly how it happens, but we know that the oxidation of the DNA goes up. So you can measure that by staining for specific markers on the DNA. Um, and oxidized DNA in lupus has been known to be a trigger for activating things like monocytes and dendritic cells and macrophages. And when DNA is oxidized, it tends to generate more interferon in a cell that takes it up, probably because it can't be broken down as easily, etc. So we know oxidized DNA is kind of toxic, right? Um, so it turns out when you UV a cell and you generate oxidized DNA, that it doesn't always stay in the structure that we had learned in basic biology, right, of the double helix.
Yeah.
Um, so the double helix is a very stable and like boring form of DNA. But when it gets oxidized, it tends to flip backwards into kind of a zigzaggy pattern. And that's what we call a zDNA or Z conformation DNA. And it turns out zDNA is actually really good at turning on cGAS. So, and the reason this is that we have another protein in our cytoplasm called zDNA binding protein 1. It's a very kind of descriptive name, but ZBP1 can bind to this oxidized Z conformation DNA. And when it does that, ZBP1 acts as a bridge to kind of just shuttle that zDNA right to cGAS and allow it to activate cGAS, which then drives the STING activation and all the downstream interferon production. So it turns out this is actually kind of a very interesting loop because in lupus patients, they have high ZBP1 expression in their skin. So if you look at keratinocytes, the cells that make up the top layer of the skin, in lupus patients, they have a lot of this ZBP1 present. And it turns out that that alone might be enough to sort of drive interferon production because when you hit the cell with an oxidizer like UV light, that will turn the process on. But the mitochondria itself has a very low level of oxidizing going on. And because of that, it's kind of always shuttling a little bit of oxidized DNA out. But normally we don't have ZBP1 around. And so that oxidized DNA doesn't really get to the cGAS. But in lupus patients, because they have a lot of ZBP1 present, they catch any oxidized mitochondrial DNA that's getting leaked into the cytoplasm and connect it to cGAS and turn it on. And so if you just increase the expression of ZBP1, you actually generate interferon production. So it seems that this, ZBP1 is a really important connector to turn on cGAS. And it also means that if we can block cGAS activity, and we show this in the paper, if you block cGAS activity, you can actually block the effects of ZBP1 overexpression in driving interferon. And so we seem to have a mechanism here that can occur either early in disease from a sun exposure, where you would generate a bunch of interferon production because of this ZBP1 that's around. And your group had work, right, that had shown that even in people who have positive ANAs, that there's a little bit of this interferon signature in the skin anyway. Um, and so that's what turns on the ZBP1. So you can imagine like this could be a very early trigger, or in somebody with established disease, you can re-trigger cGAS through this mechanism and lead to another bout of interferon spike and potentially a flare.
Which makes loads of sense, doesn't it? Because I used to think you've got to have basically what the UV does is it kills the cells in your skin, and then that's just dead cells, which is doing all that stuff that we know about. But actually,
Mm-hmm.
it's a bit more, it's a more like sensitive and finely tuned mechanism than that,
Yeah.
isn't it? It's like
Yeah.
the UV light doesn't necessarily have to kill the cell; it just stresses the mitochondria a bit, and
Yeah.
then you get the DNA in the wrong place. But unfortunately, there's also a more kind of antigenic kind of DNA.
Yeah, and I sort of equate it to going down kicking and screaming, right? So, uh,
Yeah,
you can die quietly and not cause any disruptions and, be very silent. And that's the general like cell death mechanism that we always want to happen, right? Is like it doesn't cause
yeah,
any problems. But I think UV, it does cause a lot of cell death, and interferons actually can crank up how much a cell will die. But I think what this tells us is that if you get this trigger before the death actually happens, there's like a massive alarm bell that gets sounded and releases all of this interferon.
yeah.
And then, that's going to lead to pulling in a bunch of inflammatory cells. And, some of our other work that we've done, it changes how the inflammatory cells behave when they get all this interferon, right? And
Mm.
so you change the environment around the dying cells so that when the cells come in to try to clean it up, they get turned on and get more inflamed instead of just coming in and like, cleaning up the mess and like going on about their business. So I think
Yeah.
this is important.
Even like you say, we found that that's really surprised me, but pretty much anyone who's ANA positive has a bit more of this going
Mm-hmm.
on in their skin. But for
Yep.
so their skin, their keratins are making interferon, but for some reason that's stable and it stays under control and
Mm-hmm.
it doesn't make you ill. It's just happening. But then in certain cases, something else, whether it's a more greater magnitude of that, or whether it's something else that's regulating it or controlling it, makes it turn into a systemic disease.
Mm-hmm. Yeah, and it might just be about the degree of trigger, right? Like if you get a sufficient enough trigger, like a bad sun
Strong
exposure
enough.
or
Yeah.
an illness while you get a sun exposure, there's lots of ways we could permutate this to sort of guess
Yeah.
what might be the right magic trigger. But
And the other thing is that so the skin doesn't when it starts doing it, it can kind of it upregulates itself. So it makes more as it makes more interferon, it makes more ZBP1, so it starts which makes it respond more to this path.
yeah.
So it gets one thing I often say is like because when with this focus on the skin seems to have some really important role in the innate immunity and we'll come on later to what that means in terms of therapies. But one thing I often say is like in blood immunity, everything seems to downregulate itself automatically. Like
Mm-hmm.
most cells in myeloid cells, monocytes and things, they'll do their thing
Mm-hmm.
if you stimulate them, but if you keep stimulating them, they'll stop again because they have
Mm-hmm.
and everything does. And if it didn't, if your blood didn't do that, you'd get sepsis all the time.
Mm-hmm.
Every time you had an infection. So that's you want to regulate it. But skin's the opposite because the skin's a barrier to the outside world. And so, as a barrier, you want it to keep responding all the time, every time it faces something from the environment. This is an example of that, isn't it? It's a process that won't switch off. In fact, if anything, it might get stronger.
Yeah, no, I love that
Mm.
concept. And I think the other interesting piece is that, you know, I sort of think of the keratinocytes as an immune cell in a way, in that they really are good at signaling and triggering response, like an innate immune cell, because it can trigger and signal other immune responses, right? And one of
Mm.
the things that we showed in our paper was that keratinocytes aren't the only cell type to express ZBP1 at high levels in lupus. And so we also see it in T cells and in endothelial cells and in the myeloid cells that come into the skin of patients. And so this is amplifying potentially, and we need to do more work to show this, but potentially this ZBP1 trigger can be amplified by interferon educating the other immune cells that come in. They
Yeah.
upregulate ZBP1. And so now if their mitochondria get stressed, which we know can happen, or
Yeah.
like Virginia Pasquale's work where like the mitochondria get stuck in red blood cells and then the macrophages take it up and get exposed to mitochondrial DNA that way in lupus too, right? So these myeloid cells that then are getting exposed to nucleic acids in their cytoplasm, if they've got more ZBP1 upregulated, then you can further amplify this interferon. So it's like a cascading effect.
Yeah, and in the kidney, the tubular epithelial cells can make
Yep.
type 1 interferon, and
Yep.
that's another epithelium.
Yeah.
So
Yeah.
there's a common theme, and it's and to go back to what I said at the start, is like I used to think basically the way lupus happens is B cells become autoreactive, and they make these antibodies that have a very ubiquitous target, and so you get immune complexes everywhere. But that doesn't explain why one person's got a rash and one person's got nephritis.
Mm-hmm.
Whereas actually, when you think about all these tissue specific effects, then maybe that does offer a bit more of a explanation for that.
Yeah. Yeah. I think it definitely could. I think the other explanation could just be, where the exposures tend to occur for a
Yeah,
patient, right? If things are triggered by a virus, maybe it's a systemic virus, maybe the kidney sees more of a trigger from
yeah.
that regulator, whereas the sun, I think, is just such a strong um
But you,
influencer of skin. Yeah.
yeah, but you bring in this kind of tissue responding to something environmental or some insult as a trigger for lupus, rather
Yeah.
than just saying it all starts at the adaptive immune system.
Yeah. 100%. 100%.
So basically, yeah. So I update my picture of how things happen is that the innate immunity of lupus could be coming from actually from dead or failed clearance dead cells and endosomal pattern recognition receptors like the TLRs, or it could be coming from oxidized mitochondrial DNA via the cytosolic pattern recognition receptors. But either way, we're still kind of converging on type 1 interferon between
Mhm.
those. And those pathways aren't exclusive, of course. Lots of
Mhm.
these things can be happening at the same time. And yeah, these are things that are really the common themes in all the monogenic forms of lupus. So I was thinking is, or am I right in saying all monogenic forms of lupus are somewhere around this pathway? I think it's true. I think the only one might be that the Fas ligand mutations that give you ALPS, it might be regarded as a monogenic form of lupus in sort of B cell, T cell, germinal center type stuff. But apart from that, all the monogenic forms of lupus are in this kind of very innate early pattern recognition kind of interferon part of the immune system, which has got to mean something.
Yeah. Yeah.
Yeah.
Yeah.
So, and then of course, what the other reason we're talking about all of this is because what does that mean in terms of therapeutics?
Mhm.
So I updated my like cartoon to think like where are all these different um, things working in the innate, because again, I used to naively think, innate immunity, it's all got something to do with type 1 interferon. We've got a drug for that, anifrolumab. We know it works. We like it. So you can target type 1 interferon. But actually, people are talking about things that are a bit upstream and downstream of that, aren't there? So
Mhm.
downstream of type 1 interferon, we've got JAK1 TYK2, and that's upadacitinib targeting JAK1, deucravacitinib, and some others targeting TYK2. So we've got, but then now we can go upstream of type 1 interferon. And if we want to target TLRs, there's empataran, which has some positive phase 2 data. But now we also, and this is where it gets really interesting, is we have a way of targeting cGAS sting, which is this drug developed by Ventus, right?
Mhm. Yeah. 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 It's I think really exciting because now what this is going to take us to is, really understanding the drivers of the interferon signature. And I think we're going to learn whether all patients are created equal in this regard or
Yeah.
whether there are certain drivers for certain patients. and whether targeting cGAS alone will be sufficient, or targeting TLRs alone is sufficient, or whether some patients are driven by one pathway and some patients are driven by another pathway. Um, and I think regardless of, how we sort that data out, the really cool thing is we will hopefully have ways to fix this in patients that can be patient specific and
And,
personalized, right?
and at a very, very, very high level, part of the immune process, isn't it? Is
Mhm.
like going much more upstream of the kind of the drugs of where the current drug targets work, and
Mhm.
also maybe because of that being more specific. So, like leaving other interferogenic mechanisms still working.
Yeah, yeah, which is really important when we think about side effects of interferon blockade, where we know about 5% of patients can have, you know, problems with viral infections and other things as, being on interferon blockade. And there's ways around that, right? Like we can use it to good effect, but it
Yeah.
is a side effect. And so if we can think about how to be more strategic in our therapies so that we can avoid that side effect, I think that would be really, really awesome.
So there's this therapy that's come along. It's early stage development, but it sounds like it's exciting for all these reasons because we're interested in this pathway and we can see the potential advantages of blocking it. And so there's this therapy called Vent03, developed by Ventus, which is a company I didn't know before, but they they work on this platform to develop small molecule inhibitors of these types of innate immune molecules and pattern recognition receptors, don't they? And they've developed this small molecule that can inhibit cGAS sting, and they just at the meeting in Toronto that we were all at in May presented their phase 1 data from it, and what they said was that this is phase 1 all in healthy people, not in lupus patients yet, but they had 72 healthy volunteers distributed across these single ascending dose and multiple ascending dose studies. And each cohort, they had 6 patients on the Vent03 and 2 patients on placebo and these different doses. And then they were doing PK/PD type measures. And so, because it's healthy, we don't really know what it does to the disease yet, but we know that it was, firstly, it's safe, well tolerated. They only got mild AEs, weren't dose related. Um, it's oral and it's once daily dosing. So quite nice way to give it. And I think basically we know that you get good engagement of the target, and the levels of the drug that you get in the plasma at these safe dosing levels is enough to completely block the cGAS activity. So
Mhm.
that sort of sounds promising. And I think they're about to go into a phase 2 in lupus patients, right?
Yeah, I think that's the plan.
And that phase 2 is going to be in predominantly skin disease, which makes sense given everything we've said.
Yeah, certainly this pathway is turned on in the skin. So I think what'll be really interesting to learn is um, how it modifies, skin disease itself, active lesions, but also the systemic changes that we see and whether it, you know, globally will suppress the interferon response that exists in patients, or whether it'll be a more localized change. So I think we're going to learn a ton of biology
Yeah.
about lupus with this trial as well.
Yeah. So patient subtypes who it could be good for. I mean, so the skin one's an obvious one.
Mhm.
It'll definitely work for them. But would you foresee it would work for other types of lupus apart from cutaneous?
Well, I mean, we know interferons are high in many organs, right? So, I think we don't know. What are the major driving pathways in some of these other organs? I mean, we have the advantage in the skin because we can biopsy it so easily. We can generate a lot of good,
Yeah.
basic and translational data out of the skin. I think we know that when you look at immune cells in single cell studies in like the kidney, for example, we see that they have high interferon education in,
Yeah.
most of the immune cells similar to the skin. Um, and again, whether this is a activated cGAS loop or the interferons are coming from somewhere else, I think, this is what's so exciting about this trial is we're gonna be able to
Yeah.
see, how this changes. I think they won't have kidney patients in the phase 2, um, but we'll be able to know on the blood at least if we're seeing, global reductions in interferons, and whether we see it in one cell population or many cell populations, I think,
Yeah.
will be really important questions for us to answer to then better predict who will be, which lupus patients are going to be the ones to benefit
Yeah,
most.
so
Yeah.
we know that type 1 interferons are important in really most organs you can think of that are affected by lupus. I
Hmm.
mean, I often think the neuropsychiatric aspect seems, to me, intriguing,
Hmm.
the role
Yep.
of type 1 interferon there, just because of that
Hmm.
way that, apart from there's animal models and things,
Hmm.
and we know what happens in the interferonopathies in the brain,
Hmm.
but also that, just that clinically I see these people who are clearly have pathology, but they don't have a load of inflammation.
Hmm.
And that just makes there's something going on within the
Hmm.
brain itself. So anyway, we know, or you can go around all these different organs and show that there's a role of type 1 interferon. And also we know a bit from the other therapies like the anifrolumab, the
Hmm.
JAK inhibitors, the TYK2 inhibitors, to know
Hmm.
that they are doing things to other organs as well,
Yeah,
aren't
yeah, you do see you see strong benefit in the skin, you know, benefit in the joints. I think joints are always hard to characterize in
they?
lupus
Yeah.
trials, but I do have patients who I've done interferon blockade with, and they, really have good improvement in their joint symptoms. So I think there are some patients where interferons are important there. And then certainly like all the abnormalities we see in the bone marrow too, certainly lymphopenia reverses very well
Actually,
with interferon blockade.
and you remind me of something else there as well, actually, which we kind of forgot to say, which is that so we know cGAS sting is really important in producing type 1 interferon, but it's not just type 1 interferon, is
Mm.
it?
Right. Yeah, no. Yeah, because it activates NF-kappa B pathways as well, just because of the cascade and how it amplifies things. And so, um, and TLRs are, I think, similar in this
Yeah.
regard, right? So you get multiple pathways turned on. So, I mean, the other thing that'll be really interesting to see is with a blockade of something like cGAS, whether we not only reduce interferon signatures, but we also reduce the other inflammatory components like NF-kappa B and other things that get turned
Yeah.
on when you activate cGAS sting.
With
Um,
NF kappa B being important for like other cytokines, like
yeah,
interleukin 1, for example, that you
yeah,
might.
TNF, IL-6, which
Yeah.
we
So
know are up in lupus skin and in other
and
organs
again,
as
that's
well.
another reason why this the drug target might have some differentiating effects away from other ways of targeting type 1 interferon on its own or JAK-STAT or something like that.
Yeah, no, I think it's exciting to see, how that might add extra benefit.
Yeah.
Because, I think, in clinical practice, when we're using interferon blockade, um, global interferon blockade, there's infection risk and side effects with that,
Mm.
which might also get, you know, abrogated with targeting an upstream producer of interferon because it's not the only way that interferons are produced,
Yeah.
cGAS, right? So you might have a little interferon produced during a viral infection, even if you're on something like a cGAS blocker. So maybe you wouldn't be so prone to getting, like zoster, for example.
And then the other thing it makes me think is a lot of these drugs start in lupus, but actually that's not, these pathways are important in other diseases.
Mm.
Right?
Yeah, yeah. I mean, we saw in our paper that dermatomyositis for sure has upregulation of ZBP1, and in those patients, there's a very strong correlation between the interferon signature in the skin and the presence of ZBP1. So it seems to be sort of regulated in a similar way. And we know dermato patients also are very susceptible to sunlight-induced
Yeah.
rashes. And we saw this in childhood lupus and childhood dermatomyositis, juvenile dermatomyositis. I would guess maybe even Sjogren's syndrome could potentially have links to this pathway, and
And
maybe other things outside of autoimmunity too.
I mean, there's that interesting story about this pathway being important in myocardial
Mm,
injury, post
yeah.
myocardial infarction, right?
Hmm. Hmm.
I don't know a ton about it, but just it, these because these innate pathways have a very fundamental role in response to injury. You could
Hmm.
conceive of many different roles of them, couldn't you?
Hmm.
Uh,
Yeah.
and then, of course, as always with new lupus, so as always with new lupus therapies, you get a new therapy, you've got something else in the toolkit, but now you've got a more complicated problem in
Hmm.
how to use it in clinical practice, haven't you?
Yeah.
So we're going to have to, I mean, just generally with the increasing range of lupus therapies, is we're going to have to get better at diagnosing the different immunological pathways that might be relevant to particular patients, right?
Yeah, I think that, yeah, again, and I think, this is why it's so important to use these trials as ways to learn biology and not just you say yes, no on a drug, right? Because even if the drug doesn't work, like why didn't it work or what can we learn about the biology?
Yeah,
And if it does work, like who did it work in? What cells are the most important targets here?
yeah,
So I think all of that's so important so that at some point before I retire, maybe,
yeah,
we would have a test, right? Where we could say patient X has, this this sign that they need a cGAS inhibitor, for example, and we would know that that's where we should give it. And right now our patients go through so many different drugs, like trying to find the right one that fixes their problems, right? So if we had better ways to sort of personalize our care with better biomarker testing, so we could prescribe on the first round the therapy that's best to work for them, that would be awesome. And I went into rheumatology because it was a gray area, right? Like you had to use a lot of your like immunology knowledge to sort of reason your way into therapeutic regimens for patients, right? And even though it might be more boring, I feel like wouldn't it be our dream to be like cardiology where we just have algorithms for everything,
yeah,
and
yeah.
you just follow the flow chart and you get to the answer at the end, no
Yeah.
problem.
But
Uh,
everyone who works in lupus likes things to be complicated. Why they did it. So, but here, I mean, here I suppose we started off saying like type 1 interferon signatures is quite good because that's you can say they're high or low. But
mhm.
actually now we need to go a little bit beyond that actually because you have to think where in the interferon pathway are we talking about?
Yep.
Because it's not just one thing. And
Hmm.
also, you might have to think about some of these other cytokines that
Hmm.
stratify that. So we need some other diagnostics. And then perhaps also in more pragmatic terms, I think sort of like for people who are practicing clinically now, what does all this mean? That this is
Hmm.
a drug that's not even in a started in a clinical trial in lupus patients yet. But I do think it helps just emphasize that we're there is a new world of therapy coming to
Hmm.
the lupus, and to use those therapies, we kind of have to deliver care in a bit of a different way,
Hmm.
actually, aren't we? That you need all these tools, like we said, to know, but we need also referral pathways into specialist centres, earlier diagnoses, identifying who's got the unmet need under Benlysta and things like that, who's not being served by that.
Hmm.
What are the higher hurdle targets, like being steroid free, and all of those things need to start being thought about so that
Hmm.
when these therapies come, we know how to use them and what to do.
Yeah, and I'll add one thing to that list, which is we may also have an opportunity for prevention,
Yeah,
right? So I think, because interferons are an early target, could there be opportunities here to treat very, very early and or high risk patients
yeah.
to prevent disease onset? And
Exactly, and that fascinates me too, is the idea of also not only that there might be a preventable phase of loop, like
Mhm.
because the at-risk phase, i.e. the ANA positivity, is so common compared to the established disease.
Right.
But you sort of feel like it's got an intrinsic stability to it.
Mhm.
Why can't we get back to that?
Right. And maybe
Uh,
particular aspects of these drivers might be more predictive than another, right? So
Yeah.
if you're a cGAS activator, are you more likely to develop lupus than if you're, a TLR7 activator? Like these things can be sorted out and studied, and I think that's going to be
And
a
the
really
things
important
and
next step.
the drugs you might use to prevent lupus might be slightly different from the ones you choose in established lupus because it's a different. Okay, I think we've probably run out of time there, but that's a really good place to finish up. So thank you again for your time. And to the listeners, thank you for listening in again today. And if you could subscribe to this podcast on Spotify, iTunes, or other podcast media, let us know what you think because we do listen to the feedback in deciding what we're going to do next time. And you can watch this and other podcasts on Lupus Forum on YouTube as well. And if you follow us on X, LinkedIn, or Facebook, then you'll get updates whenever something new comes out. So thanks again.
Thanks, Ed.